Dear students, today we are going to learn about growth curve of bacteria. That means when we explore microbial growth patterns in a controlled condition, we can see a similar pattern or we can get a graph. So, we can, so students, what is a bacterial growth curve? It is a graphical representation of bacterial growth, population growth over time under controlled condition. Note this point under controlled condition. It tracks how a number of variable cell changes as bacteria is introduced into new environment. Okay, is it doubled or declined? That in the such case, such details are can be explored from this growth curve. The curve reveals how bacteria multiply, how means its speed or frequency, and adapt to the new environment and respond to new environmental factors such as nutrient availability, temperature, and the accumulation of metabolic waste products. These all the factors how affect the growth of bacteria. We can explore from this curve. By studying this growth curve, scientists can be predict bacterial behavior and make informed for the decision making in medicine production or food safety or in case of industrial bioprocessing like waste management, all that. You can move into growth curve. There are four phases for the growth curve. The four phases are very important. So you must learn all these phases without confusion. The first phase is the lag phase. Lag phase. This graph represents the x-axis is cell count and y-axis is time. Time. This is cell count. See this lag phase. This is the first phase. This is the phase of adaptation. Adaptation. See, we can see there is no increase in cell count during the time period. So this is lag phase, lagging the cell growth. In this stage, bacteria get adapted to the environment, a new environment. This phase is called log phase. That is second phase, log phase, also called exponential phase or rapid phase of rapid division. Log phase or exponential phase or a period of rapid division. This lag phase and log phase will not get confused. Okay. The second phase is called log phase or exponential phase. In this phase, this is the phase of rapid growth or rapid multiplication of cells. Cell division is in as a fast and the cell count gets increased. Okay, see, we can see the graph um, go upward. That means cell count is increased. And third phase is called stationary phase. As name indicates, it gets static. Number gets static. No changes in the number of cell count and number of bacteria. That means no further divisions happened there. This is called propagation, stationary growth. Growth plateau is seen, called as a growth plateau. And fourth phase is a decline phase. That means death phase. Death phase means the number of the cell count is declined. Number of bacteria is declined. That means we already know the replication is stopped. The multiplication of cell is stopped. Then what happened? And that means the death of bacteria is remained bacteria get died and cell count is declined. Cell count is decreased. The curve below illustrates how population changes over time under control. This is the graph. This graph represents or shows how the bacteria's growth is happening in a controlled condition. And second, we can see characteristic of each phase. First one is lag phase. Lag phase, as we say, bacterial is adapted to new environment. Is become adapted to new environment. So there is no bacteria is even the bacteria is active, but no further multiplication is happened. Second phase is log phase, that is also called exponential phase. This is a phase of rapid multiplication or rapid division of cell bacteria. Okay, and population is increased or doubled because of abundant nutrients and optimal condition for growth is here. Third phase is stationary phase. Stationary phase growth rate is low and nutrients depleted and waste accumulated in this case. So cell division is slow O, cell division and cell death reach equilibrium. That also is that may be the reason for the uh, stationary or graphical plateau formed there. That may be the reason. Cell division and cell death is become equal. So no increase in this graph. Okay. So this keep population stable, and in this case, waste accumulates, nutrients depleted, and cell division and cell death is equal, and population is stable in count. Last phase, death phase. In here, cell death exceeds the new multiplications of cells or new formation of cells because of the exhaustion of nutrients in the environment. We already said that it is a controlled condition, a controlled environment. So the nutrients is exhausted. So no further multiplication is happening or cell death is exceeds the new cell formation. So graph is declined, decreased. Cell count is decreased, and graph is declined to out downward. Total viability of death cells is increased. Death cell is increased. Viable count of viable bacteria is decreased, and death is increased. Okay. This is the graphical representation of cell growth, microbial growth curve. So this graph is, you should learn this graph thoroughly. It is very important to draw the graph in an examination. So this is lag phase. I'll didn't repeat again. This is log phase and this is stationary phase. This is the phase of decline or death phase. What should be the significance of growth curve from the industry, medicine industry, or pharmaceutical industry, or food industry, or food products, or waste management? There are all the like the uses of growth curve. By using growth curve, scientists or engineers can understand the behavior of bacteria in different conditions. So they can explore the behavior, how to respond the bacteria, how will respond bacteria to this environment or to this medicine. In this condition, they can learn or they can explore by using bacteria. There are I given here 4 points about the uses of the growth curve. It means it helps to determine optimal timing for antibiotic treatment. Okay, in what time period or what time the antibiotic will respond or will decline or destroy the bacteria, they can learn from this growth curve. And also, it is critical for the fermentation of and bioprocessing industries, fermentation of food products, and also bioprocessing means waste management or other uses. And also, it provide insight into in bacterial behavior and physiology. It is useful when we learn about the new bacteria or new organisms we found out. And it's useful in food safety and microbial control strategies. It is useful in food safety and microbial control strategies. And we can form new strategies to control microbes in foods. Or it food causing microorganism causing food spoilage. And it is useful, thereby it is useful in food safety. And thank you, dear students, for listening this presentation. And further questions, you can message me.